I want to thank the organizers for both inviting me to present today, but also just putting together this seminar series. As you'll see, we're kind of new to the sphingolipid field. We've studied amino acid and fatty acid metabolism for many years, and sphingolipids are just kind of a great direction for us to go due to their involvement in both pathways and their biological activity. So today I'll talk about, for the most part, published studies on how amino acid alterations and SPT promiscuity impact cancer progression. And if there's still some time at the end, I'll touch on some of our work on the neuropathy side of it, which correlates quite well. If there's two take-home points to really think about, the first one is somewhat obvious, but sphingolipids are tightly and physiologically linked to amino acid metabolism, and this manifests itself in certain disease states. And what we're starting to see now, by manipulating and controlling sphingolipid biosynthesis or turnover, we can break this link. So my lab has strong interest in applying stable isotope tracer technology to measure metabolic fluxes. Fluxes describe the ultimate function of a metabolic enzyme and its physiology, and that in some cells and tissue cases is a phenotype in particular. So this is where the chemical engineering and analytical chemistry meet cell biology and genetics. An important point to think about in how non-scalable flux measurements are, they require multiple measurements of analytes, some spatial components, a time component, and even a cell biomass component to do it properly. Stuck here. So what we try to do is use stable isotope tracers and mass spectrometry to quantify enrichment in a localized or kinetic manner to capture the spatial and temporal aspects of metabolism. Why my pointer's stuck? So, and that's the general approach we take in my lab. We apply stable isotope tracers to different biological systems, ranging from tissue culture samples to animals to patients, use mass spectrometry to quantify metabolite abundance and isotope enrichment, and use some fairly simple, the simpler the better modeling to interpret these data and resolve flux changes in the metabolic state of a system with the hopes of identifying mechanisms and potential targets. We have a number of different interests. Cancer metabolism is our main focus. We study diabetes, lipid physiology, as well as some nervous system disorders and retinopathies as well. There's one common theme that's emerging from a number of these studies. It's that we see metabolic flux and enzyme promiscuity has an impact on the lipidome. So in the case of the study today, the origins come about 10 years ago from two studies that were published, and one of which I was peripherally involved in, that demonstrated that PHGDH, the rate-limiting step in serine biosynthesis, is amplified in triple-negative breast cancer and melanoma. The serine biosynthesis pathway is known to be upregulated in a number of tumors, and dietary deprivation of serine and glycine, because they're highly exchanged, has been shown to synergize with chemotherapy and slow tumor progression. This is data I'm showing right here from our lab. So the questions that have remained that we've tried to answer is, why are serine and glycine so important for tumor growth, and what are the molecular mechanisms that drive this sensitivity? And I highlight the plurality there because it's in biology, it's never just one thing that's happening. The first question is really challenging because serine and glycine are important for a lot of different things metabolically. As we'll hear about today, serine is precursors for certain lipids, protein, of course. It's highly exchanged with glycine in one-carbon metabolites, whereby it impacts redox, bioenergetics, methylation, and nucleotide synthesis, as well as glutathione synthesis. So there's a lot of different pathways. So in thinking about this question, we tried to think about how cells control flux through different systems. because we have enzymes that are tuned to channel substrates to specific pathways. So we went to the Brenda database and pulled the Km values for different serine utilizing enzymes that are in the cell. And these are listed here along with their names. And just to remind you, the Km tells you the affinity that an enzyme has for its substrate. And notably, so the lower the Km, the higher the affinity. And what you can see, if I overlay here, if we also look at the concentration of a xenograft tumor in a mouse fed a serine replete diet or a serine free diet, we see a range of about 0.5 to 1 millimolar in terms of the concentrations that we see. So what this means is that the enzymes to the left of this blue bar should have plenty of serine available to them and will have no problem binding it. And this makes sense when you think about it, because protein synthesis, phosphatidylserine synthesis, and nucleotide redox metabolism are critically important for cells. If you can't synthesize protein, a cell is not going to survive or change its state. These enzymes here on the bottom to the right-hand side tend to be more tissue-specific, more monofunctional. Serine palmitoyl transferase seems to be in the sweet spot, with a caveat of how hard it is to accurately measure a Km of a ER-associated enzyme. This really suggests that serine levels in the body are physiologically tuned to impact flux through serine palmitoyl transferase, suggesting that moderately low serine, which is what we observe in vivo, could drive alterations in sphingolipid metabolism. This community has fully appreciates the importance of serine palmitoyl transferase, but I would just hold it's important for producing signaling molecules and structural membrane lipids, but there's also a highly active salvage pathway where lipids and sphingosine from the membrane, from S1P, can be salvaged back into the ceramide pool, such that active SPT flux is not always needed for cells. What's also been learned over the last decade is that serine palmitoyl transferase is a promiscuous enzyme, whereby it can use either alanine or glycine instead of serine. These lipids lack the hydroxy group here, so they can't be used to synthesize more complex sphingolipids or S1P. So deoxysphingolipid itself was originally isolated from a marine clam and actually shown to be toxic to cancer cells and administered to some patients around 2000 or so, but those trials were halted due to liver toxicity. About 10 years later, a number of labs demonstrated that deoxysphingolipid was an endogenous molecule. This is in particular led by work by Florian Eichler and Thorsten Hornemann, the latter of whom have really laid out the synthesis and degradation of this class of sphingolipids. We and others have also observed that wild-type SPT in the context of low serine levels in patients with a macular disease, Metallo, will also drive accumulation of deoxysphingolipids. So the question we wanted to ask is, can we somehow coax tumor cells to make deoxysphingolipids? And while this makes sense to some degree based on the serine results, it is a metabolic correlate. So we actually came at this and more from an alanine angle at first, based on some discoveries in vitro. So when we got started on this path, we were actually trying to understand how metabolism is altered when cells grow under anchorage-independent conditions. This I'll call it spheroid growth, is a hallmark of tumorigenic cells. And what we do is plate cells on low attachment plates and so they can't adhere, and they have to start from single cells and grow out to these spheroids. And one of the things we immediately observed was that in all cases, despite the relatively slower growth rate of cells in spheroid conditions, their alanine efflux, the alanine secretion flux, increased dramatically. almost twofold. And this is a twofold increase in a flux, not a metabolite pool. Metabolite pools go much larger. There's some other impacts as well on TCA intermediates that were indicative of mitochondrial stress, and we worked out those details in the manuscript. But what we also saw was an accumulation of sphingolipid bases, in particular deoxysphingolipid, if we hydrolyzed the cells and looked at the total accumulation of sphingolipid bases. So this alanine phenomena was interesting to us, in particular because of prior work we did studying the mitochondrial pyruvate carrier. What we found is if we inhibited the mitochondrial pyruvate carrier or knocked down the genes encoding this, what we uniformly saw in cancer cells was a decrease in alanine levels. And this is shown here. You can see that adherent to spheroid increase in alanine efflux. When we treat with an MPC inhibitor, alanine levels and alanine secretion reduced. And what we were able to show is happening is when you block the MPC, the cells would use a bypass mechanism and bring alanine and oxidize alanine in the TCA cycle. What we were also able to see, and which was observed by others in the field, if we inhibited MPC, it has no effect on adherent growth, but it would dramatically increase and improve the spheroid numbers that were generated. As it's been demonstrated that MPC is downregulated in colon cancer and its overexpression will reduce spheroid growth. So this presented us with the interesting question, is alanine deleterious for spheroid growth? It's actually one of the most abundant amino acids in our blood, in our tissues. Plasma concentrations are approach 0.5 millimolar. Yet most tissue culture media don't have it at all, and they're high in glutamine as the primary source of nitrogen. But in fact, if we culture cells in the presence of supplemented alanine at 1 millimolar, we would see a reduction in spheroid growth, and this was to some extent improved by MPC knockdown. At the same time, we observed if we inhibited the MPC or knocked down the genes encoding it while increasing spheroid growth, the deoxysphingolipid levels in these cultures would drop significantly. So again, this is exciting and demonstrates that the alanine levels really correlate quite well with deoxysphingolipid levels, but it doesn't really functionally connect the two. So what we next observed, if we treated cells with myriosin, this is a classically used SPT inhibitor, and typically it's used in publications at 1 micromolar. I've seen as high as 10 micromolar. But what we saw is even at moderate concentrations, adherent growth was unimpacted. And when we got to higher levels, we would see a slight reduction in growth rate. On the other hand, spheroid cultures would improve in the presence of myriosin from anywhere between 1 nanomolar up to 1 micromolar in concentrations. So we confirmed, we tended to settle on 10 nanomolar, and we validated that even at 10 nanomolar, we see a pretty strong inhibition of flux through serin palmitoyl transferase as measured by 13C serine incorporation into sphingolipid over time. What was really exciting, if we look at the ratio of deoxysphingolipid to sphingolipid in molar terms, we see a dramatic accumulation in spheroid culture, but that ratio would be diminished by inhibiting SPT. So if we bring this together, what we were able to actually see across a number of different colon cancer cell lines, that in all of these cases, myriosin enhanced spheroid growth, alanine supplementation diminished it, and in the presence of the alanine, myriosin would rescue and mitigate those deleterious effects. And as you can see here, with the alanine supplementation dramatically increases deoxysphingolipid levels, and this was suppressed by myriosin treatment. And we've seen similar results using fumonisin B1 as well, because deoxysphingolipids are known to be toxic only when they're converted to ceramides. So this really highlighted that spheroid cultures are sensitive to DOXA production, And we wanted to know why, so I'll share some unpublished work here that kind of gets at that question. What happened if we look at the spheroid culture system as starting from single cells, the establishment of small spheroids to larger spheroids, and broke this down into different temporal parts? What we were able to observe is when treating cells with either an MPC inhibitor, alanine, myriocin, or fumonisine B1, it induced the expected effects. But if we waited till day 3 to do these treatments, then we had no impact. So really what we're starting to think more about now is these deoxysphingolipids as a sensitizer to membrane-associated stress, which of course occurs when you trypsinize and plate cells at single cell. And those stresses combined impact and drive this spheroid phenotype, because these lipids aren't as toxic under adherent conditions. So this is all good and well for spheroid growth cultures, but what happens if we go to the in vivo system? I will remind you, serine glycine-free diets are very different than serine glycine-free culture media, as the diets, the non-essential nature of these amino acids allow the animal to kind of maintain reasonable levels of serine and glycine. And what you can see in the tumors of these, in the xenograft tumors of mice fed a serine glycine-free diet, we saw elevated alanine levels and about 50% abundance of serine and glycine. But deoxysphingolipids dramatically increased. What I'm going to show you here, and mind the axis labels here, what if we total the different deoxyceramide, ceramide, and dihydroceramide species, you can see dramatic accumulation in both the tumor and the liver of these mice. But the fold increase in the liver is much less than what we observed is significantly less than what we observed in the tumors, suggesting that tumors have this intrinsic drive to synthesize deoxysphingolipids, depending on the amino acid environment, and potentially a reduced ability to degrade the compounds. But it is a good reminder, especially from a location standpoint, that metabolite changes in response to diet are very tissue specific. I won't go much further down this pathway now, but what we did see, well, we didn't see a change in canonical ceramides in the tumor or liver of these animals. We did see a reduction in some abundant glycosphingolipids. And this is one avenue that we're trying to develop and pursue a little bit more now, particular based on a lot of the great talks that we've seen in this seminar series. But it really highlights that amino acid restriction alters membrane lipid composition. So we took a shot at also rescuing growth on a minus serine glycine diet, and interestingly observed that only at low doses could we rescue spheroid growth. And in both cases, we saw a moderate reduction in deoxysphingolipids, but interestingly, the big difference came in the case of the liver. A high dose of the normally used dose of myriocin, it dramatically suppressed liver ceramide levels. Those mice lost more weight, and the tumors were not really able to grow really well. And this really highlights the fact that canonical sphingolipid synthesis by the liver as well is important to support tissue growth and tumor growth. So separately, we also tried to target the serine synthesis arm. We obtained an inhibitor of PHDDH and administered that to the animals prior to inoculation with the xenograft tumors. The inhibitor reduced serine and glycine levels in the tumor and in the plasma, and induced sphingoid base accumulation. So just to quickly sum up on the tumor side here, what we've seen is that amino acid metabolism impacts the tumor lipidome. And we've seen this by manipulating serine availability, mitochondrial pyruvate flux, SPT flux, and PhD flux. That said, most of the serine that's synthesized in the body is actually in the nervous system. If you can see this by infusing an animal with isotope labeled glucose and looking at enrichment of serine across different tissues. And just shown here in red, these are all nervous system tissues or parts of the eye. And this has been known, and glial cells, astrocytes, are the main synthesizers of serine in the nervous system. But this was intriguing to us because we were also starting to investigate a disease called macular telangiectasia. When I moved to San Diego, my neighbor actually was in charge of is still in charge of this Metallo project, and he brought us online because they had made some interesting discoveries whereby genetic hits in the serine biosynthesis pathway pointed to serine potentially being lower in these patients. And in work that we weren't involved in, they observed that serine and glycine were down in the blood of these patients, suggesting that Metallo is a serinopathy. So we then looked to quantify deoxysphingolipids in these patients and saw, when controlling for various factors, Metallo patients had significant increases in deoxysphingolipids along with the lower serine levels. And if we just looked at the broader population, controls and Metallo patients, we see a really significant and tight correlation between serine levels and deoxysphingolipids, as well as alanine levels, which correlate positively. What was also interesting, we had diagnosed several patients with Metallo that actually had hereditary sensory and autonomic neuropathy type 1, the disease I mentioned before, whereby SPT LC1 or 2 mutations drive deoxysphingolipid synthesis. And you can see that these patients are outliers in the correlations. More interestingly, if we went to our mouse model and extended the time that we applied the diet and fed a minus serine glycine-free diet for as much as 10 months, we could see thermal hypoplasia induced by this serine glycine-free diet, as well as ERG defects, which is exciting because we're now seeing that the environment can be a driver of disease phenotypes, vision defects, and pain sensation, in the same way that genetics can drive this, which is observed through mutations in SPT LC3, some coding variants, and GWAS hits in PHGDH in the case of Metallo. But at the same time, you also see some genetic events in cancer that drive this effect. So it's exciting to be able to see the same biochemistry manipulated both through environment and genetics. So just to quickly sum up what we've discussed, deoxysphingolipids, they seem to compromise anchorage-independent cell growth while having no impact on adherent growth. We think this is a combination of deoxysphingolipid accumulation and membrane-associated stress, suggesting that these lipids could be a signal about membrane health. There's a number of different potential targets that have implications in a range of diseases. Outstanding questions remain: why are deoxysphingolipids toxic? And what effects do they have on complex sphingolipid metabolism and associated processes, some of which we'll hear about in the next talk. So quickly want to thank the folks that did the work. Selvam Muthusamy, he's now moved on to industry, led the cancer sphingolipid work, and the Metallo work was a great collaboration that we have with the Lowy Medical Research Institute and Marty Friedlander and others. Lastly, I'll put a plug in. Here's a picture of the beach yesterday in San Diego. It's a nice place to quarantine or socially distance. We're looking to expand on analytical and cell biology fronts in a number of arenas, so please shoot me an email. Thanks a lot for your time.